So without further ado, I'd just like to introduce Yuri. Yuri was awarded a PhD 9 years ago in March 2013 at her university in Japan. And her PhD was about studying the physiological basis of butterfly colour vision, and supervised by Professor Kentaro Arikawa. As a world-leading vision scientist, he introduced Yuri to the importance of understanding how animals perceive the world using comprehensive and precise methods, from molecular to behavioural. When she finished her PhD, to our benefit, she came to Australia and did her first postdoc work with Professor Jan Hemi at University of Western Australia. And they looked at the colour vision in both nocturnal and diurnal Australian bull ants and the visual system in fiddler crabs. So this is diversity, isn't it? Then she moved to Macquarie University in 2016 to work with Dr AJ Narendra's lab, which was furthering the work on visual navigation and eyes in Australian bull ants. And then South Australia. So Yuri came to work with Professor Karen Nordstrom, who we've heard of here, to understand the motion vision in hoverflies. Yuri says that since she was little, she's always wanted to understand animals and diversity in behaviour, body shape, colour and eyes. During her PhD, she worked on Colias erate, a sister species of the cabbage-white Pieris rape. She was fascinated by the fact that they can see a broader spectrum range than humans. Besides, she enjoyed catching them around her institute almost every day, where she could see Mount Fuji across the ocean and raised more than 500 butterflies to use in her experiments. Her research aims to understand how animals see their world with limited but specialised sensory systems and neurons in a tiny brain. So thank you for coming, Yuri.
Hi, thank you very much. Kind introduction, Jerry. Hello, everyone. My name is Yuri Ogawa from Flinders University. So this is my picture of butterfly today. We got this. I took car park 1 in Flinders University, and there are milkweed bushes there. So if you would like to see them, please come along. So today I'm going to talk about, it's a real honor to me to talk about butterfly vision today. And I'm so excited we explore together how to see the world. And I'm gonna just minimize this for a while. How butterflies see the world. So we are going through, we are going through 3 main topics today to understand how they see the world. So first of all, I will introduce briefly vision in animals. And then I got detail about what's color and color perception. Thank you. To begin with, I would like to show some beautiful pictures. They are all eyes in different animals. You can see plocked larvae, the top, this one. And then flatworm, they do have very simple eyes. They just detect the light direction. And Nautilus, they do have slightly better eyes. They can sense, they can see, they do have some spatial resolution to see the object shape. And the beautiful blue dots. They are eyes of scallop.
Wow.
They are very unique because of the looking, but also they use a mirror system to focus the light onto the retina instead of having lens. And sea fan also have a light sensitive structure. Oh, sorry. Those bands are light sensitive structure of sea fan. And then we do have different compound eyes in krill, dragonfly, lobster, lens, and mantis shrimp. And especially in mantis shrimp, they do have 12 different color channels in their middle of their eye. You can see the bands. They can see color very well. And then we do have octopus eyes here, and spiders have single lensed eyes. Okay, so now we kind of see the diversity of eye shape, but functions are also very different. And then I can say vision is a very crucial sense of in many animals, and then their tasks are specialized for ecologically relevant behavior for each species. I would like to talk a little bit of example, the specialized task in hoverfly. So hoverfly is a very good flyer, but also they also very good to detect the moving objects. So hoverfly, which I'm working on at the moment, and you can see hoverfly is hovering in midair, and as soon as he enters into the other male's territory, he is detected and chased away. So from the resident territorial male's perspective, he is seen as a very small moving object, but the pursuer can perform this behavior with specialized eye and neurons. I wouldn't go too much detail about the neuron today, but I would like to highlight that male's eye are specialized to detect motion, detect the moving object. As you see in the compound eyes in males are joined at the dorsal part, but not in females because females are not territorial. Okay. Wow. Did I convince you? Yeah. Okay, then I'm going to move on to butterflies. I'm showcasing Japanese butterflies. In Japan, we also have blues and swallowtails and eastern pale-clouded yellow Colias erate, which I worked on during my PhD. And we do have skippers and some graphium, and then also some nymphoid butterflies. And as we all know, butterflies are magnificent. Butterflies have magnificent colored wings, and they can visit different colored flowers. So it's easy to say butterflies must have a good to detect colors. Yes, it is. It has been already demonstrated in early 20th century by Dora Ilsa in Germany. She used butterfly, and she also used the artificial colored flowers, and then to test whether they can learn the color or what kind of color they prefer. And I'm going to show some videos that she filmed in 1970. So you will see the feeding butterflies here. So when feeding, during the feeding, butterflies extend mouth part proboscis, and that behavior can be used in her experiment. So here she used white cabbage butterflies. And now she is showing different color patch among the different gray scale. But butterflies prefer going to the yellow patch. So it means butterflies can see the color based on the color, not the light intensity of the patch. So the white cabbage butterflies can prefer purple, and then also you will see they visit to the red as well. Shall we? Yes. This is really fascinating. Yes. And then even they didn't give any sugar water, butterflies can extend their proboscis to show the feeding behavior. So this can tell us they can learn the color. Okay, so after her experiment, late 20th century, my PhD supervisor, Michio Kinoshita and Kentaro Arikawa's group did the similar experiment in papilio, swallowtail butterflies. And then, as you see, papilio can learn yellow patch, and then they can discriminate from different gray scale of the patch. So it means they learn yellow, not the gray light intensity of the patch. So now I'm going a bit detail of what's color. So our eyes or animal's eye receive light, and light is a wave. And then color is determined by the length of the wave. So wavelength is shorter. It can be seen as bluish, but wavelength gets longer. That looks like as a reddish. Okay. And in nature, the color is created by absorption and reflection from the object. So we see the leaf as green because the pigments on the leaf surface absorb all spectrum except the green. So we see the leaf as green. But if you see the bananas, we see as yellow because they are reflecting on the yellow spectrum. But we see the white paper, it's because white paper can reflect all spectrum of the light. And absorption and reflection is the one way to create color in nature. The other way, another way is structure of color. So this is an example from bird-wing butterflies. As we all know, they are very beautiful, but their color, yellow to blue, are not created only by the pigment, but also the structure on their wing. And then when we closely look at their wing, you can see scales, as you know. And then when you cut through one scale, you see you see this kind of repeated structure. And then nanoscale structure of this can create orange to blue color difference. And then structure correlation is not very only for butterflies, but also many different animals. For example, peacock spider and jewel beetles, and then many of birds, and then some species of butterflies pupae is very closely shiny. That's because of the structure made by the structural color. And then they use this structural color for communication. In peacock spider, only male have that beautiful coloration, and then to show their conspecific female for mating. And then structural color can not only create the color, but also create very dark black. So this is an example from butterwing butterflies. And you see, as I told before, the green part is created by repeated structure on the green scale. But you see their black part is so dark, and then that's called as ultra black. And then that darkness is 10 or thousands darker than charcoal or so on, like normal black. And then that's because it's not created by pigment, but created by structure. So when we look at the scale and magnify it and go in detail, you see lots of holes on the scale. And then that hole is very deep and then thick. So light traps in the hole cannot go out, so that in the end, the light trap there, and that's why we see so dark. So we go through what's color now, and then I would like to go a little bit more detail how to see the color. I want to start with our color because it's kind of good to understand too. So in our eye, there is a retina back of our eye, and where we receive the light. So in our retina, we have 4 different types of photoreceptors, and one is rod, which is functional during the dark. So at night we use rod, and then we have 3 cone receptors: blue, green, and red. And cones are for color vision for us. So at night we only use rod, one photoreceptor type, so we can see the color at night. That's why. Okay, and then we have in our retina, we have blue, green, and red receptors to receive the color information. And the information from the eye goes to your brain, and it's around here. And then the brain creates the color based on 3 different color channels. So most of us have trichromatic vision. So that makes us easy to see the red object, ripe fruits, for example, on the tree with 3 color channels. And if we have only 2, we see the same picture as this, so it's hard to see the red fruit anymore. And if you have only 1 color channel, all comes to monochromatic. So having multiple color channels are quite important to see the difference in color. Now I would like to do some exercise with you. So I put some animals picture, including my little cute cat and my friend's dog. So I would like to ask, who has trichromatic vision? Do you have any idea? Would you? So who do you think gorilla has trichromas?
Yeah.
And how about male New World monkey? Okay. Do you think cats have trichromacy? No. And how about female New World monkey? Yes. And how about squid? Yeah. How about shark? No. And dog? Okay. All right. Thank you for answering. Yeah. So answer is only gorilla and some species of New World monkey, but only for female. That's because the color genes to create color receptors are on the X chromosome, and then so that's why. So if you have two X chromosomes, you have a high chance to get more color channel. So now I slide down. So trichromatic vision can be seen in gorilla and some female of New World monkey. And then who has dichromatic vision? That's you may know the cat's dog and then male New World monkey, because they only have one X chromosome, then they highly likely they cannot get extra genes for color channel. So the answer is this.
Wow.
Yes. That's a very good question. Thank you for asking. Yes.
Repeat the question.
So I have a question. Where the bird fits in this figure? That's very good question. Thank you. Because I didn't put the birds in there because they do have tetrachromacy way beyond us. So it's already known that birds, fish, and reptiles can see UV in addition to blue, green, red. So it's very fascinating. Yeah. Okay. So the reason why most of mammals cannot see even like trichromacy is they are originally nocturnal. So they kind of lack the ability to see the color. So that's the reason, the evolutionary reason why most of mammals cannot see the trichromacy. Okay, and this is the artistical illustration, how different vision we have from birds. So for us, it's not just but a beautiful bird, but yeah, still blackish, but bird could see the pattern like this. So birds are very good. And then not only the color vision, birds are very good to see the fine detail. So the y-axis is showing the acuity, which is spatial resolution. So like how well they can discriminate. So you do eye test, right? And when like I don't have good acuity, so I cannot read these letters on paper. When even I don't know how to say that. This even so like acuity is saying spatial resolution. Is that right? Okay. And then, so high acuity means they can see the detail well, and low means they cannot see the detail very well. And then human sits around the blue arrow here, but birds is better than human. And then insects, including butterflies, sit way down, so they do have lower spatial resolution. Okay, I'm going back to insect now. So insects have 3 different color channels, but they are different from us. They can see UV instead of red. So their color vision is shifted towards the short wavelengths side. But this I worked on when I was in UWA, and then it's quite fascinating because they are nocturnal, but they can see color in the dark, having 3, because for human, we only have rod, can be functional during the night in the dark. So we cannot differentiate the color at night, but they can. So honeybee has also UV blue-green, and most insects have UV blue-green. And I just compare what kind of range they can see as color is, as I said, human can see blue to red, but most insects can UV to yellowish. So when we see the yellow flower, we perceive as this, but for insects, they can see as this because, color isn't correct, but in the center of the flower, they often have UV refracting part, which is called as nectar guide. So you can see as a black pattern. So they can see that pattern very well because they can see UV. So I said, this is from most insects, but we have gone through that. As we have gone through that, butterflies must very good to detect color. So what are butterflies? So this is my favorite butterfly, Colias erate, and you can see their face as this, and they do have big enough compound eye, and one compound eye consists of 6,000 of a unit called as ommatidia. And then as we talked about the acuity before, they do not, so each ommatidium create 1 pixel of image, so which means they can create only 6,000 pixels image. So it's quite rough image they do see. So if human would have the compound eyes become like this, 1 meter in diameter. I love this picture. So going back to compound eye, this shows the compound eye structure much more detail. So as we could see here, I hope you can see some facets, dots. That's the lens of each eye unit, ommatidium. And then, so in each eye unit, there is a lens, and then photoreceptors underneath of the lens. So light comes through the lens, so it's like a straw structure, packed straw. So light comes through the lens, can only perceived by the photoreceptor underneath. So light cannot go through from here to there. So only light can travel through the only this part. And in butterflies, in butterfly eye unit, there are 9 photoreceptors, and then 4 at the distal, we call distal, but 4 photoreceptors at the distal, and then 4 proximal, and 1 in the basal part of the eye unit. And then when I cut through the eye horizontally, you can see the eye like this. So can you notice 4 reddish dots in each ommatidium, sorry. And then their distribution patterns are slightly different. One is like this. I often think this is like a pig face, and then some square, and then rectangle one. So we can, I wouldn't go much detail, but there are 3 types of ommatidia in one butterfly eye. So I said 9 photoreceptors are in one eye unit. And then now I would like to know what kind of color channel are in one ommatidia or in one eye. To understand that, we can do electrophysiology experiment. So I immobilize one butterfly here. They were alive. And then I cut a very tiny hole on the cornea to penetrate glass electrode to record electrical activity of single photoreceptor. So I shined different color of light and then see what kind of response I can get from one photoreceptor. And then response, you can see the response gets larger and larger when I show them blue light. So I can tell now, yes, that photoreceptor was blue receptor. So and then I stained that photoreceptor to localize in one eye. Yeah. So that's the example. So that blue receptor was the larger photoreceptor in pig-like face ommatidium. And then like doing this kind of experiment 300 times or more, I finally could make this figure. Yay! Thank you. So we found that there are 8 types of color channels in both male and female. 1 UV, 2 blue, and 2 green, and 3 red receptors. So as you could notice that butterflies can see UV to red range, which is nearly like about birds, and then much more than most of insects as well as humans. Yes.
It's interesting that the female seems to be able to discriminate the reads better than the male.
Yes. Yes. The question was why female can see the red range better than male. And that's I'm going to talk. Yeah, thank you. So why female have different peaks in red receptors? This we found the reason in their eye. So this is again, when I cut through the eye horizontally, we see the reddish pigments. And then in male section, we only see red in different shape. But in female, I hope you could notice the orange one is existing. And also the type 3 one, like a rectangle one, is closer together. And then what they are doing in the eye is they are act as an internal sunglass. So, which means photoreceptor, so only red light pass through the pigment can be received by the photoreceptors. So those pigments screen the all spectrum except red, so that orange one create orange, orangish red receptors, but like a rectangle closely together one has a stronger effect of the sunglass. So therefore they can create the deep red receptors. So orange, red, so and the dark red. And then why female needs to have them. And this is still our speculation, but because they are sexually different, so we are thinking that's for example, female specific behavior such as laying eggs, and then to find the good quality of the leaves of the host plant, which is clover for them, they may use the multiple red receptors. Okay, so now we know that the Colias erate is belong to Pieride. So this is a phylogeny of butterflies. And then Colias erate, which I worked on for the PhD, has 8 photoreceptors. And then we also know how many photoreceptors in sister species, white cabbage butterfly. That's the 7 close. And then they can also see from UV to red range. And we also know some swallowtail, how many color channels in swallowtails. And then Papilioxutus have 6. And then 4 out of 6 photoreceptors are used for foraging behavior, which is tested already by behaviour. So this is a bit tricky, but like how electrophysiology is good to understand the physiological mechanism of color vision, but we also need to test behaviorally how exactly which types of color channels is exactly used for each behavior. Yes. So that's a bit different. So they use tetrachromacy at least for foraging behavior. And then we do know graphium. They do have 15. That's the 15 is the largest number in nature we know. And then how do you think about skippers? Skipper is locating in between swallowtails and Pieride. Would you think they do have a lot? But not. They only have 3. And their eye structure are also different. Like I didn't tell much detail, but most of butterflies have a position compound eyes, which the light cannot travel across the eye unit. A superposition eye, to increase the sensitivity of the eye, eye can travel across the eye units. And then skippers have superposition eye. And then other species do have tetrachromatic eye, UV to red. So very complicated. And then we still don't know how this diversity occurs yet. But my collaborators and I am interested in some nymphalic butterflies have only 3 receptors. That's we can see in Nymphalini, the Vanessa, so on. And then they are in the same subfamily. So if we compare between them, we might be able to understand how natural selection, how nature creates this diversity in butterfly. And then one possibility, the color and complex color vision may relate to the finding behavior. For example, some nymphalidae butterflies perform patrolling. So male going around the field and try to find the female who could be feeding or who could be laying eggs, or maybe not feeding, yes. The other species perform perch, which is male do not go around. They do have territory and sit on the branch or perch and then wait, passing through the female. So in that case, if that's true, patrolling species could have better color vision because they need to discriminate female wing coloration at the leaf background. But for perching species, color cannot be very important. Rather, they could spend energy to detect the moving object. So this kind of project I would like to do near future. So finally, I would like to thank my supervisors, Kentaro and Michio, who did behavioral and then physiology and molecular work together with me. And then Douglas de Wenger in Netherlands. He is very big boss, but he is very kind and to teach me how the red pigment works in the eye. And then I also worked with Yang Hemi and RJ Narendra and Martin Folksky and Johan Zell to understand the blue ants and the Fidra crab vision. And then I'm working with Karin at the moment. And then for the future study, I would like to work with Primoz to understand how complex color vision has been evolved. Thank you very much for your attention.
Okay. All right. Thank you very much for those wonderful questions, both here and on Zoom. And thank you very much for those people on Zoom for persevering with us. We had 24 people on Zoom. It was really great.
Oh, yeah.
So I know that, Yuri, you've probably got a copy of our book, but this is a special one with the signatories of our authors, apart from Lindsay. But I'd like to accept that special copy.
Yeah, thank you.
Thank you very much.
Thank you very much.